Hello friends, I am Ayantika. In this video, I will be discussing about the overview of Chara, its thallus structure, its ecological importance, and in the next part of the video, there will be reproduction in Chara. Now, Chara, it has got certain taxonomic importance because though this Chara was earlier considered as a chlorophyte, but now it is considered as a sister group of chlorophyte because Chara has many characteristics that are close to embryophytes, that is the land plants. In fact, this Chara, they are considered as ancestor of land plants because the charophytes, the cell division of charophyte is just like the cell division in land plants. Both land angiospermic plants and the charophyte, during cell division, the microtubules, they lie parallel to that of the direction of the spindle. Whereas in chlorophytes, when the cell plates are organized, the microtubules, they are arranged perpendicular to that of the spindle. So in this way, the charophytes are much closer to the embryophytes than to the chlorophytes. So we can easily say that this Chara, it is an ancestor of this land plants. And also Chara has similarity with bryophytes, like Chara also contains lignin and sporopollenin. This presence of lignin and sporopollenin makes this Chara resistant to desiccation. So we can see that Chara has got a great taxonomic importance also as a algae, apart from its ecological importance. So let's start the video. Before I start the main video, there is a quiz for you. This is an easy question. If you know the answer, then write it in the comment section, or otherwise I will be discussing it at the end of the video. So, which of the following algae is found to be growing on the fins of certain fishes in Sweden? It is the fin of fishes, not the scale of fishes. I've also some videos back, I have also put a quiz like which algae grows on the scales, but it is not here. I have not given the scales, but I have written fins of certain fishes, so be careful to answer this one. Overview of Chara, or it is also called stonewort. First, we discuss the taxonomic position. It is belonging to division Charophyte, class Charophyceae. All the Charophyte or the algae belonging to the Charophyceae, they have their the cell wall. They have calcium and magnesium carbonate in their cell wall, and that is why it is very rough to touch. The rough to touch is due to this deposition, and it is common in all the charophytes. And this charophyte, they are also called Carassian algae or stonewort due to this same reason, the rough to touch reason. And the charophytes, they are mostly occurring in very clean water, and they grow both in fresh water and brackish water, but it's always depending upon the species. Same species won't be growing in the fresh water and the brackish water. So different species will be growing. The freshwater species are different, and brackish water charophytes are also different species. But whether they grow in freshwater or brackish water, they prefer very clean water, shallow water, to maximize their photosynthesis. They prefer shallow water and clear water, and the water should also be very not very much flowing. The flow rate should be very low. In India, actually, this the all the charophytes, they are preferred. They are seen growing during the winter months, which suggests that these charophytes, they prefer growing in in lower temperature. So next is the order Chara. Now order Chara includes also the main feature of the Chara is that they have a thyroid body. The thyroid body is mainly divided into branched structure. The body is divided into node and internode, and the main axis as well as the side branches, they have a lateral whole of branches. This characteristic of Karalis is very unique. That is, from the nodes of both the branches as well as the main axis, a whole of short branches appear. So this is the main characteristic of order Karalis. Now, family Karasi. Now, Karasi, it includes those algae where the body is the main thallus body is divided into nodes, internodes. The body is branched, and the chloroplast, they do not have pyranid. However, starch is the reserve food material, but they are not stored in pyrenoids. They are stored in the root in special structures called the bulbils or the amylum starch. So, this absence of pyrenoid is a main characteristic of Charophyceae. And also another important characteristic of order of family Charophyceae is that their sex organ. Their sex organ are very specialized and multicellular. In fact, the sex organ of the Charophyceae family, they represent resemble the land plants in their complexity, the reproductive structure. So this is all about the taxonomic position of genus Chara, the algae we are discussing today. Its habitat is submerged. It is always submerged, though it can survive desiccation, but mostly it grows in submerged. The it can it is found to be growing luxuriantly during the winter months, and the summer month they die or they perinate as bulbil, myelum starch, or like this type of structure. Perinating structures are present here. So the habitat is and that they are submerged. They are found to be growing both in fresh as well as brackish water, and they are actually benthic algae, so they grow on soft mud at the bottom, or they are attached to certain, they grow epiphytically, and these they prefer clear water and slow moving water. They can grow even in alkaline limestone area water also. Alkaline, one of the alkaline species is Chara baltica, and it can also grow in hot water, but not all species, but only Chara fragileus, it can survive in hot water. Otherwise, most of the Chara species, they are cold preferring algae. So this Chara, they grow aggressively in absence of angiospermic plants, because Chara cannot tolerate shade, and if there is no angiospermic plant, then Chara can utilize the sunlight totally. There is no shedding by the angiospermic plant. So in absence of angiospermic plant, Chara becomes very invasive. And if there is angiospermic plant, Chara won't be able to survive much because of the shading effect. So this is the how the invasiveness of Chara is dependent upon the presence of other species, especially the angiospermic plants. So, out of the 180 species in India, only 27 species has been noted. So these are the common species of Chara that are found in India. These are the common species. So Chara gives a very musky odor due to the presence of sulfur when it is crushed. And the color of the Chara is usually green, but when it is desiccated, it can look a bit brownish also. And it is cosmopolitan in distribution. Mostly it is found in the temperate region in the northern hemisphere, and in India they are found to be glowing luxuriantly during the winter months, but during the summer they die out, and they perinate in the form of bulbils or amylum starch, it depends. And these are the perinating structures. So the vegetative thallus of Chara is found only during the winter month. So this is about the distribution of Chara. One thing I should be saying that apart from growing as attached to the substratum, it can grow epiphytically on the other algae as well as other angiospermic plant. Chara, it grows mixed with Tolipella. This Tolipella is another charophyte, and also it can grow on Ceratophyllum, Myliophyllum and Eloidea. Okay, these so Chara grows along with other charophytes, both submerged as well as grows epiphytically also. One thing I should be saying that Chara itself hosts another algae, that is the Coleochaete, inside its body. And this Chara brownie, it is an endangered species, considered endangered in Poland. The most of the reason behind endangerment of Chara is that though it can survive high temperature in the form of perinating structure, and it can survive alkaline water, but it cannot survive shading and muddy water, because Chara requires very clear water for photosynthesis, and also it cannot grow in very contaminated water. It requires very clean water. That is the main reason. The pollution, water pollution, is the main reason for the endangerment of various species of Chara. Thallus organization of sandgrass. Chara is also called sandgrass due to the rough texture. It usually grows to the height of 20 to 30 centimeter, but depending upon the type of species, it reaches 90 centimeter also. So the plant body, it looks like this, which resembles superficially equisetum or the hostel. So it resembles the land plant, and the entire thallus, it is divided into stem-like root-like structure. So it is attached to the substratum by rhizoids, which are branch structures, multicellular branch structures, and haline, that is, they do not have any color. And the upper portion, that is the shoot-like structure, is divided into node and internode region. This is the internode region. This entire is the internode region, and this is the node region. In the node region, we see that there arises a whole structure, whole of branches. These are called the primary laterals. These whole branches, these are called the primary laterals, or branches of limited growth, or branchlet or leaf. Why they are called branches of limited growth? Because depending upon the type of species, it usually grows from 5 to maximum 15 internodes, and after that it stops. So these are all about the primary laterals. And from the axis of the primary laterals arises the branches. These are the branches. These branches, they are called the long branch or auxiliary branch, because they can grow very long. And this primary lateral, they arise from the division of the nodal cell. But these this auxiliary branch, they arise from the division of the internodal cells. So the branches of unlimited growth arise from internode cells, and the branches of limited growth, that is the primary laterals, they arise from the nodal cells. So this is about the branching. Then as we zoom on to the secondary laterals, to the primary lateral, this one, we see that the primary lateral, or the leaf or branchlet, what we say, it is divided into nodes and internodes. We know that 5 to 15 nodes or internodes will be present. So at the nodal region, we have stipulodes. Stipulodes are also called the secondary laterals. Here we see, this is a very difficult to see here, but in this picture it is very clearly shown that they are arranged at the nodes in a hold structure. And at the axle of this stipulode, or the secondary lateral, arises this sex organs. Here the upper one is called the nucleus, which is the female sex organ, and the lower one is globule. And I will be discussing the detailed structure of nucleus and globule in the second part of the video, when I will be discussing the reproduction. So for now, just remember this nucleus and globule and the structures. So this is the furthermore zoomed picture, where we see that the main axis, this is the main axis, this is the internodal cells, and this is the node from where arises this primary laterals, and from the base of the primary laterals, this we see this stipulodes, which are also called. These are the points that I was discussing in the earlier slide. So this the nodes. Uh, the nodes in there is a difference in the cellular organization in nodes and internodes. What happens is that in the node has two central cells and 6 to 20 peripheral cells. And this is what happens in the nodes, and the cells of the nodes are isodiametric. But the internode can contain either a single cell, or it can contain many cells surrounding it. Now of these cells, if it is single, if the internode, they have a single cell, then it is called E-corticated. E. Here I am denoting it with just E. E-corticated. This type of internode is called E-corticated. If the internode has a single cell surrounded by many cells, then only it is called corticated. Now there are many species of Chara which are corticated and E-corticated. This is the corticated Chara, and this is the E-corticated Chara. Now we see that the corticated Chara is Chara zeylanica, and E-cortication is seen in Chara brownie, Chara wallisiana, Chara corallina, Chara brownie, and Chara nuda. So this is about how the transverse section of the node and internode looks. So this is all about the dimorphic branches of limited growth and unlimited growth that I have discussed in the earlier slide. So the whole of branches, they can be 6 to 16 depending upon the type of species. So the secondary lateral or the stipulodes, they are bistipulode, these are the species Baltica and Tomentosa. Unistipulode, that is they are arranged in a single layer. The bistipulode, it is the two layer. This is the two layer around. This is the main axis. So this is the main axis, and these are the two layers of stipulodes in bistipulate, and unistipulate has only one layer. Rudimentary stipulodes are also present, that is in Chara, Wallichi, and sometimes stipulodes are also absent, that is Chara partially. Now, growth in Chara is mainly by the division of apical cell. When we have discussed Oedogonium and Eulothrix, like this type of algae, we see that growth was in the intercalary cells and the cap cells. But here in the Chara, growth occurred by division of the apical cells. And then the rhizoid, so already I have told that rhizoid is multicellular, but one thing is that they have oblique septations. That is, oblique septations means that this is the normal septation. This is the cell where the septation is complete, and oblique septation is that the septa formation is not complete. So in root, this is the diagrammatic representation of how the nodal and internodal cells, they differ in their basic cellular infrastructure. This is the nodal cell number A, and this is the internodal cell. Here we see that the nodal cell, it is isodimetric in shape, and this internodal cell is elongated. So the nodal cell, apart from its shape, it is also different in size because it is much elongated, much larger. And also it has got a single nucleus, but it has got the internodal cell, it has got many nuclei. And also it has got a very large vacuole. This is the vacuole. Whereas in the nodal cell, it does not have any a few vacuole, or sometimes vacuole are also absent. So how does the nodal cell differ from that of internodal? Number one point is that the size of vacuoles, the number of nuclei. Apart from that, there is also third point is that the cytoplasm. The cytoplasm is divided into dense ectoplasm and less dense endoplasm. The cytoplasmic streaming is found mostly in the endoplasm. So this is the ectoplasm. The outer layer, it is the ectoplasm. We write it with capital. The ectoplasm is, we write it with capital E. This is the ectoplasm. We write it with capital E. And here no streaming is observed. But in the endoplasm, that is, this is the endoplasm. This is the endoplasm where we observe cytoplasmic streaming because it is less dense. So there are three different this is all about what I was discussing in the previous slide. It's just you can just stop the video and just read it out. I'm not reading it out because I already discussed. function of Chara. Now, Chara, just like other algae, it stabilizes the bottom sediment. So, in that way, it reduces the turbidity of soil. At the same time, it also acts as a food for a wide range of organisms. That is, from macro to micro invertebrates, from the prey fishes, as well as the waterfowl. So it is acting as a food for a great variety of organisms. At the same time, it is also the habitat of many organisms. So basically, they are of many function creating a clear water habitat, and also food for the aquatic organism as well as the organism that mostly stays in water, like the waterfowl. So it has got great ecological significance, but it has got also certain research significance also, especially this Chara australis, this species of Chara, it is attacked by tobano virus. Now this tobano virus, it infects the land plant also. So if we want to have research work done on this tobano virus, then what we do is we cultivate this Chara australis and inoculate the tobano virus in the Chara. So by this way, the research work on the tobano virus will be very easily done, because the Chara, it is thalloid in structure, and so any algae, because of their thalloid structure, it is more easy and simple to study and the metabolic functions than that of the land plants. So this is the research importance. Also, Chara has got allelopathic substance. Now these allelopathic substance, they exclude limnatic snails and phytoplankton also. Also, where Chara grow, we do not see any mosquito larvae. This is also due to the allelopathic substance. The Chara does not allow growth of snails, other phytoplankton, as well as the mosquito larvae due to this allelopathic substances. Chara has got certain a negative impact also. Like for example, if there is a eutrophication, then Chara can go luxuriantly. But also sometimes it happens that if the water body in which Chara is growing is surrounded by agricultural land, then the agricultural effluent will include herbicides. So these herbicides will again kill um, the angiospermic plant. So say for example, this is a pond, and all the surrounding area has agricultural land. So during the rainy season, what happens is that all the water accumulates in the pond containing the Chara, and this effluent will include the herbicide that were used to check the unwanted weeds. These herbicides or these weedicides that were used in the agricultural field has now landed in the water containing Chara. Now Chara being an algae, it won't be affected. Only the metabolism of the the angiospermic plant will be affected by this herbicide or weedicide. So in that water body, there will be no angiospermic plant. So in absence of angiospermic plant, there will be luxuriant growth of Chara. How? Because Chara is very sensitive to shading, and this shading was caused by the growth of angiospermic plant. Say for example, nymphia, it has got big leaves. So this nymphia or nelumbo, so if these plants, they die due to this effect of herbicide or weedicide, then only under that condition, in absence of shading, Chara will grow luxuriantly. So we can see this rapid colonization of any water body by Chara It is an anthropogenic or human caused effect. And also Kara has another important function, that is it harbors cyanobacteria that grows epiphytically on it. So this cyanobacteria, as we know that they are producing cyanotoxins. So this cyanobacteria, if it grows luxuriantly on the Kara surface, can it can cause in long term it can cause the cyanotoxins will be released when the cyanobacteria dies, the cell wall burst. So in water containing Kara, and if that Kara has a surface layer of cyanobacteria, then that water can become toxic also. So these two are the negative effects of having Kara. Now we come to the quiz part. Which of the following algae was found to be growing on the fins of certain fishes? Here we see there are four options: Chlorella, vulgaris. Chlorella is found to be growing on the scales but not on the fins of fishes. Then we have this Ectocarpus fasciculatus. This is a marine algae that is found to be growing on the fins of these fishes. So the option is option B. Option B, Ectocarpus. And Spirogyra condensa and Coleochaete nutella. The Coleochaete nutellarium, this species of Coleochaete is found to be growing inside the Nutella. Nutella is another charophyte just like the Chara. So this Coleochaete nutellum is a found to be growing endophytically on this Nutella. So here the answer of this quiz is Ectocarpus fasciculatus, option B, that is found to be growing on the fins of certain fishes. Soon I will be making videos on Ectocarpus. Earlier I have made videos on Volvox, Pseudogonium, Ulothrix, Chlamydomonas. So I have put all the videos that I have made in the description box. I have put the link. So you can easily click the link and watch those videos. With this I end my video, and the first part of my video, that is the overview of the overview and the thallus structure and the ecological economic importance of Kara. In the next part I will be discussing about the reproduction. In any case you want me to make any video on any other subject, you can easily write it in the comment section, or if you want me to do any more topic in Kara, then also you can write it in the comment section. Also, if you have any suggestions, do write it in the comment section. Thanks for watching this video, and if you like it, share it with your friends, and please do subscribe my channel. Thank you.